pharmaceutical stabilization of drug substances against chemical degradation. The headline: stabilization by modification of molecular structure of drug substances, stabilization by complex formation, stabilization by the formation of collagen complexes with cyclodextrins, stabilization by incorporation into liposomes, micelles, or emulsions, addition of stabilizers such as antioxidants, and stabilization through the use of packaging. Let's start with microbial degradation. Contamination in the introduction of microorganisms into product. Contaminating organisms may arise from many sources during the course of manufacture and subsequent use of the product. Spoilage is followed by the contamination and describes the processes and consequences of the microbial growth in the product. Microbial spoilage can be caused by bacteria, yeasts, or fungi. Effects of microbial contamination: infection hazard, loss of potency or effectiveness, unacceptable to the patient or consumer, toxic effects. For instance, endotoxins produced by gram-negative bacteria, change of activity, the inactivation of biological active molecules by organisms contaminating a formulation. Destruction of penicillins by the penicillin's enzymes, loss of atropine in eye drops by Corynebacterium and Pseudomonas cpp. Visible effects: in liquid formulation, contaminants may be seen as a sediment, turbidity, or polycal, while on more solid preparations, colonies, often colored, of bacteria, yeasts, or molds may form. Color changes. Color changes due to alterations in the components of a product may result from pH redox or other changes caused by the metabolic activities of an organism, or to pigment production by the contaminants themselves. Gas production: bubbles, frothing, product containing carbohydrates or other fermentable substrate, or particularly susceptible to this type of spoilage. Irritancy, especially in eye preparations. Olfactory effects: production of characteristic odor, for instance sulfur containing metabolites such as hydrogen sulfide, fishy odor of amines, and the astringency of ammonia. Texture: creams can become lumpy or gritty, and changes in viscosity of liquid preparations. Sources of contamination: the raw materials, including water from which the product is manufactured, the manufacturing environment, including the atmosphere, equipment, and work surfaces, manufacturing personnel. Control contamination and spoilage during manufacture. Standards for atmospheric cleanliness in the manufacturing areas for the production of both sterile and non-sterile products can be followed. Air supply can be invariable HEPA filtered cleaning and disinfection procedures should be followed properly. Raw materials may be exposed to radiation, ethylene dioxide, to reduce high level of contamination. Filtration units and UV light can be used to remove contamination in water. If the water is to be used as an ingredient for injectable product, filtration is preferred over UV light. Water can be maintained at 80 Celsius degree whenever possible during manufacturing process in order to prevent microbial growth. Preservatives should be chosen carefully during formulation for the medicinal products. The properties required in preservatives: a broad spectrum of antimicrobial activity covering gram-positive and gram-negative bacteria, yeasts, and molds, and no chances of resistance development. Low toxicity for humans, so that can be used in topical, oral, and parenteral products. Good solubility in water, stable and effective over a wide pH range, and compatible with common excipients. Non-volatile, odorless, and tasteless. Selection and use of preservatives. Parabens are used in topical and oral products. Eye drops contain benzalkonium chloride as preservatives. Examples of preservatives: ethanol, isopropanol, propylene, also propylene, glycol, and glycerol, as shown in this chart. Preservatives used in pharmaceutical preparations, preservative and concentration, percent, weight, volume. Injection, for instance, preservative phenol, cresol, chlorocresol, the concentration as shown here, 0.5, 0.3, 0.1. Eye drops, chlorhexidine acetate benzalkonium chloride 0.01. Mixtures: benzoic acid, methyl paraben, alcohol, and as shown here 12 to 20 the concentration of alcohol. Creams, parabens, chlorocresol, as shown here the concentration. Tablets, methyl paraben, also 0.1 concentration. Prevention of microbial degradation, suitably designing the containers, add antimicrobials as preservatives, single-dose containers, sticking to proper storage condition. Stability testing. To provide evidence on how the quality of a drug substance or drug product varies with time under the influence of a variety of environmental factors such as temperature, humidity, and light. FDA stability studies include testing of those attributes of the drugs that are susceptible to the change during storage and are likely to influence the potency and safety. Testing should cover the chemical, physical, biological, and microbiological attributes, preservative content, for instance, antioxidants, antimicrobial, preservative, and functionality tests, for instance, drug dissolution tests. The stability needs to be evaluated over time in the same container closure system proposed for marketing. Frequency of testing: long-term stability testing every 3 months over the first year, 0, 3, 6, 9, and 12 months, every 6 months over the second year, and annually thereafter. Accelerated stability testing minimum of 3 time point, for instance, 0, 3, and 6 months. Intermediate stability testing as a result of significant change at the accelerated storage condition minimum of 4 time points, for instance 0, 6, 9, 12 months. Significant change for a drug is defined as failure to meet its specification. If no significant change occurs during the 6 months accelerated and real-time stability testing, the product will be allowed to place in the market with a provisional shelf life of up to 2 years. Case notice this chart: case, study conditions, and minimum time the storage. Now, case general study long-term conditions. 25 Celsius degree to 2 Celsius degree, 60%, 12 months, and the humidity 5%. Intermediate 30 Celsius degree to 2 Celsius degree, 65%, 6 months, humidity 5%. Accelerated 40 Celsius degree to 2 Celsius degree, 75%, 6 months, humidity 5%. Stored in refrigerator long term, 5 Celsius degree to 3 Celsius degree, 12 months. Intermediate 25 Celsius degree to 2 Celsius degree, 60%, 6 months, humidity 5%. Stored in freezer long term, minus 20 Celsius degree to 5 Celsius degree, 12 months. Stress testing. Using exaggerated storage conditions to increase the rate of chemical degradation or physical change of a drug product help identify the likely degradation products, useful in establishing degradation pathways, developing validating suitable analytical procedure, to evaluate the effect of short-term excursions outside the label storage conditions. For instance, high temperature during shipping, temperature in 10 Celsius degree increments, for instance, 50 to 60 Celsius degree above that for accelerated testing. Humidity more than or equal 75% relative humidity HR. Thanks for listening.